So our results from our gel electrophoresis. So sample 365 and 373 had very weak small bands, as well as the negative, and the positive had some strong bands since it had E. coli in it. So the quantification of our amplicon PCR, so in our sample 373 and 365, there wasn't DNA in range, but we had some in our positive control, and the concentration of that was around 15, and the amount of the buffer was 90.24. We also found that there was DNA in our negative control, which there was not supposed to be any DNA because we never put any in it, but the concentration was about 0.535 and the amount of the buffer was 77. Sorry, so for our reads, so we had to choose different samples since they weren't processed in time for us to read our results. So we changed this to the samples 307 and 343, which had similar characteristics. So the total number of reads and successes for 307 was 3,023, and the amount for our second sample, which was 343, was 2,687. So the diversity and relative abundance of bacterias that we found in our sequencing and possible controls was, so we basically made a few different graphs. We made one for kingdom, order, and genus, and we labeled the X as the organism detected and the Y was the percentage. So for the kingdom, it shows the top 2 of the 8, and sample 307 had a high percentage in bacteria and less percentage in unclassified. So for our order graph, it showed the top 3 of 8 classifications. So Clostridialis, unclassified, and Bacteroideus are the classifications in that chart. Sample 343 had a high percentage in unclassified, had a higher percentage in unclassified than sample 307 had. For our genus graph, it showed the top 3 of 8 classifications, and Clostridialis, unclassified, and Bacteroideus are the classifications in the chart. Sample 343 had a higher percentage than sample 307, because it had a low percentage of those 2 bacteria. So the categories other and unclassified, so they mean 2 different things. So the other category, it's reads that they can be identified, but they were found in less than 3.5% of the total reads, and they can be classified as considered unimportant. However, the unclassified category, which were found in all of them, are categories that they don't match any known organism within that level, which means that might most likely resulted in errors in the reads. So a discussion of our DNA extraction from scat. So the DNA extraction wasn't completely successful in terms of quantity because it was very low, and our DNA, so in general, DNA concentration has to be between 250 and 260 in order to have enough concentration to track, and if there's more than that or lower than that, there's likelihood that there was some kind of cross-contamination or the DNA was contaminated. And specifically for us, our UV spectrophotometer results really showed that was the error that we had. So regarding our amplicon PCR results in the gel electrophoresis, is that they were unsuccessful, most likely because when we were pipetting into the wells, some of our samples leaked into neighboring wells, thus contaminating our results. And we were mainly able to see this due to the fact that of all of our samples, the ones with the highest percentage of DNA was our negative control, whereas the other ones did not have enough. So we're pretty sure that was the cross-contamination that we had. So talking about our quantification results of the indexing PCR, so the results were very low concentrations of DNA from all of our samples. there was, as I said, DNA in our negative control, and our positive control had some, not as high as our negative, but it did have some results from the E. coli. However, we did expect that our negative would have 0. But like I said, it turned out having DNA, and that indicated our cross-contamination from our samples. So discussing the number of reads of the positive control. So most organisms found in our samples were unidentified organisms. So it was about like 0.5%. However, Blottia hydrogen trophica was an identified organism found in our samples at about 5%. Blottia is a bacteria found in normal gut microbiome in eukaryotes to process sugars like lactose or fructose or glucose, and even undigestible materials such as plant matter. So another identified organism found in our samples was bacterioloids, and they made up of around like 12% of our findings, which was pretty significant. And they're bacteria that are commonly found in human intestines, and they insist in the breaking down of foods. But they can, if they're introduced to other parts of the body, they can cause infection or disease, and just have negative feedback. So what our results tell us about the cross-contamination is that it happened somewhere around the middle of our processing DNA concentration. And there's a probability that we either didn't listen to a specific step, or by accident we mixed up a sample, which resulted in our inaccurate results of DNA. So discussing the number of successful reads in each of the animals compared. So there were no successful read amounts in the animals that we compared and did analysis on. There wasn't a high enough concentration to get a proper read of any of our samples. So discussing the differences between the gut microbiomes of the group of animals that we chose and comparing them to the kingdom, the order, and the genus, as we did here. So bacterioloids are a genus that we found in both of our animal subjects and in different orders. Sample 343 had bacterioloidicase at 20%, and sample 307 had the same bacteria at about 30%. Bacteroids in sample 307 had 12%, and at the genus level in sample 343, there was around 7% of bacteroids, zilacibolids, and bacteroids were around 2%. So there was a very small difference between the bacteroids at the genus level, but they are classified into different orders. We couldn't find details on specific orders of the bacteroids, and a site that we used was MicrobeWiki. So addressing our question, was that we were going to analyze the gut microbiome in 2 dogs under the age of 10 months of different genders, to see whether the gut microbiome in males, as we hypothesized with females, might have better gut microbiome than males. So our data did not support this hypothesis because our results were skewed. Also, our samples that we tested didn't come back, so we had to use other samples. And in our original samples, our DNA concentration was too low to gather adequate results to support or deny our hypothesis.